Good morning, everyone. This will be the last micro lecture on this developmental origins of health and disease, and I thought that I would like to introduce at least the concept of epigenetics and epigenetic mechanisms as being in the origin of the developmental origins of health and disease. And you see, it's not that we have many examples where we have actually proven the existence of epigenetic mechanisms, but as time goes by, this evidence will be increasing because there is a lot of people that's actually working on this topic. This epigenetics is basically appears to be the ideal mechanism on how to explain the existence of fetal programming mechanisms. The idea here is that these epigenetic mechanisms impose changes at the genetic level that can be inheritable. There's a lot of debate about that, but at the same time that they can explain the sort of secret appearance of these disease mechanisms. Because the idea here, remember, is that a fetus exposed to certain conditions that may not be that optimal trigger the appearance or the increased appearance of diseases later in life. But you could say that for most of the period, I mean, when basically this organism is a baby, is a child, is a young, there's nothing apparently wrong. It's simply that later in life, some of these diseases kick in and increase in probability depending on the conditions during the fetal period. And epigenetic mechanisms seem to be ideal because basically they appear later on, but they are silent for some period of life. Essentially, there is lots of potential periods in which epigenetic changes can take place. If you go from gametogenesis to the time of conception, so that this is basically the production of oocytes or sperm, the conception, the fertilization, birth, weaning, growth, maturation, reproduction, and aging, there is basically evidence that epigenetics can have a role in many of these different stages, as we will see in a second. Epigenetics, if we have to define it in a way, has been defined as the branch of biology which studies the causal interactions between genes and their products, which bring phenotypes into being. The idea here is that it's no longer in the DNA that the changes are occurring, but are changes above. That's the idea of epigenetics. It's not in the sequence of nucleotides that appear in the DNA, but essentially modifications that we will see in a second, in the way that this DNA is existing and that this DNA is allowed to be expressed. And this is a nice cartoon on how these epigenetic changes actually can take place. You know very well that chromatin, or that the DNA is compacted in chromatin, and there are proteins called histones that help in this compacting. We know very well that certain chemical modifications of histones, in this case it's showing acetylated group, acetylation in the histones, can actually affect the degree of compacting of the DNA and be affecting gene expression. This is actually controlled by this enzyme histone deacetylase. We know also of the existence of other epigenetic changes, such as the presence of methyl groups in different DNA bases. This is what appears here. And when these methylations appear particularly in the promoter region of certain genes, there may be a silencing, an increased or a decreased, usually a decreased expression of these genes. So in the example here, if you have this particular gene that's characterized by the promoter region and different exons that are going to be transcribed, what you have is the normal transcription event and the synthesis of protein. However, if this promoter region is actually methylated, the idea is that it impedes methylation, impedes the attachment, the binding of transcription factors, and by not binding, basically this gene is silenced. So essentially, epigenetics goes down, at least we know most about DNA methylation methylation on cytosines, but also other modification on the histones that can actually be taking place. We know very well that if we go back here, important changes, important methylation changes take place very early on. And what this is describing is two major periods of epigenetic reprogramming. One is actually taking place in the germline. That means the gametes, that means the oocytes and the sperm. The second one is the one that's shown here. It's a post-fertilization large epigenetic reprogramming in the form of methylation. What you see here in this graph is basically as developmental time proceeds, you have a degree of methylation from a low methylation to a high methylation. This is the mature oocyte that basically has been fertilized. And notice that very quickly after fertilization, there is actually a decrease in the methylation levels in the oocytes, or in this case in the zygote. There is a decrease in the methylation levels, and then at some time later, there is a new methylation appearing here. This happens also earlier during the gametogenesis. What was puzzling about this is that people really knew about this for a long time. So basically, it's very clear that large methylation changes occur after fertilization. And what this actually told or suggested people is that epigenetic marks are not inherited. Epigenetic marks are not permanent, because at least twice, very early on in the formation of the organism, methylation disappears. There is a tremendous disappearance of methylation changes, which then reappear again. Nowadays, we know that the picture is more complex. First of all, some genes stay methylated. This is this line here. These are called the imprinted genes. Imprinted genes are those genes that basically are expressed only from the paternal or the maternal copy. Imprinted genes means that very early on in the development of this zygote, there is one of the copies, one of the alleles that is silenced. It can be the parental, it can be the maternal. And that's what's called imprinting. Imprinting means that out of the two copies of a gene, one is silenced very early on. These are imprinted genes. Imprinted genes basically, or we know that imprinting actually occurs by methylation, and this is a permanent mark. This does not disappear. But for most of the other methylated genes, the methylation marks disappear and reappear again. We now know also that with some mechanism, basically this methylation can be placed back in the same location. So to some extent, there is an inheritance of the methylation locations in different genes. Even when the methylation drops and then increases again, methylation marks can be placed back in the same location. And that's what explains the transgenerational effects that have been observed with these epigenetic mechanisms. As I said, some genes that we call the imprinted genes are not modified, but most genes actually are. Some epigenetic markers are transmitted to the next generation as well. What evidence do we have that epigenetics is at the base of some of the fetal programming mechanisms that we were talking about? And in this case, one case that has been studied very well is the Dutch famine cohort that I was describing yesterday. In this case, a particular gene, or an important gene in growth is actually the insulin-like growth factor 2. That has been known for a long time. IGF-2 is key in human growth and development. We know that IGF-2 is a maternally imprinted gene, means that only the maternal copy of the gene is expressed, while the paternal is suppressed. Imprinting in this case is controlled by methylation, and if you have a hypomethylation of one of the alleles, basically you have biallelic expression. So in most people, you will see imprinting of the gene only the maternal copy is expressed, but if actually that is affected somehow by a decreased level of methylation, you have both alleles that are able to express this gene would no longer be imprinted. We also know very well that the methylation mark on IGF-2 is actually stable up to middle age. So if it's affected early development, the effect will be seen many years later. So here we have a candidate gene, IGF-2, that we know is imprinted. Only the maternal copy is usually expressed, and the methylation marks are stable. So what people have been interested is in going back to this Dutch famine cohort and looking for some or looking for the patterns of methylation in IGF-2 in the individuals from this population. What have they found? Well, what they have found is the following. This is a paper by Heymans et al., published in the Proceedings of the National Academy of Sciences in 2008. And they've done a nice study. They've done a nice study, but this graph is a little bit complicated. Let's see. First of all, you have this data is shown in terms of date of birth. When the individuals of the Dutch famine, or some individuals, the ones that you have here in this dot plot, basically were born, and notice, and this is something we know relatively well, that the ratio, the amount of food that these people was receiving, or the mothers in this case, the mothers of these children were receiving, stayed very low from December 1944. And then, of course, when the Allied forces basically took over again and liberated Holland, around May 45, the ration increased because again there was food available. So we have a well-characterized period of malnutrition. If we now look at the date of birth of these babies and we look at the methylation patterns of IGF-2, what is it that we see? Well, in the y-axis you have basically the percent difference between exposed and unexposed siblings. To make it more complicated, not more complicated, basically to adjust for individual differences, what the researchers have done is take one child that was, or the child, member of the cohort that was born in this period, in these dates, and then make a ratio between this child and one brother or sister of him that was not born during this period of malnutrition. So that would be your control. For every single child of the cohort, you take a control sibling and then look at the expression, at the methylation of the IGF-2 gene in both and do a ratio between them. What do you notice here? Basically, 0 would show no difference, right? 0 would say that there is no difference in the methylation pattern between the cohort child and one of its siblings that was born outside of the malnutrition window. Down to 0. Basically, all this data clusters around 0. There is no difference. So, when you look for differences based on the date of birth, you see no differences. That means that children that was born within this period from mothers that were malnourished do not show evidence of different methylation patterns here. The interesting thing comes in the next slide. In this slide, you do see a difference. You do see a difference. Basically, the exposed children, the children from this cohort that were exposed to malnutrition, show now a lower degree of methylation. But notice that now we are not plotting this graph with birth date. What are we plotting it with? Last menstrual period of the mother. Because the last known menstrual period of the mother is the best indication that we have for time of conception. For time of conception. So what this data shows is that if you look for the time of conception, presumably within 1 month from the last menstrual period, or you could say a couple of weeks, when you look at from this, you see a consistent pattern of hypomethylation in the cohort child. Not when you look for birth of date or birth date, But when you look for the last menstrual period corresponding to the conception time, you see a difference. What does this mean? May I remind you the concept of critical windows we were talking about yesterday? So what this is suggesting is that when conception occurs at the time of malnutrition, you have effects. In the period graph, the child was conceived quite a few months ahead, earlier, right? Basically, you know that it takes 9 months for a baby to be born. This is June. So if this is June, you have to go back to when? October. And that's, in this case, the latest children here would have been conceived in October, late September. And that's exactly the period before the malnutrition period started. So, in other words, malnutrition in this case is very important if it occurs around the time of conception, but not so important if it occurs around the time of birth, or when the mother is late pregnant. Does it make sense? Right? So I think that this is a beautiful evidence that actually epigenetic markers play a role in the effects of fetal programming that we have been talking about. And to me, it's the most clear example, but I expect that in years to come, we will be seeing differences both in experimental models and also in human cohorts. And that's the end of it. Thank you very much for it. Time for questions.